This video is brought to you by Saraswati Vidyalaya Union. And hi, this is Prapti. Today we are going to study about the second step of aerobic respiration from chapter Respiration, that is Krebs Cycle. In the last lecture we have seen about the glycolysis. That glycolysis was the first step in the aerobic respiration. Now let's study in detail about the second step, that is the Krebs cycle.
Now, let's study about the Krebs cycle. This Krebs cycle is there. This Krebs cycle, which is the second important step of this aerobic respiration. This Krebs cycle, which is discovered by the scientist Sir Hans Kreb in 1937. That's why this Krebs cycle, which is known as the Krebs cycle. And these scientists, he discovered this Krebs cycle in the pigeon's muscle. For his discovery, he got Nobel Prize in 1957. Okay, as well as this Krebs cycle, it is also known as the citric acid cycle. This is known as the citric acid cycle because in this, the first compound which is forming, that is known as the citric acid. As well as this Krebs cycle, which is also known as the TCA cycle. In most of the organic acid, so there is a presence of 3 carboxylic acid group. That means COOH group. So these 3 carboxylic groups are present. That's why this is known as the tricarboxylic acid cycle. As well as this cycle, which is known as the organic acid cycle. This is also known as the organic acid cycle. Now, why it is known as the organic acid cycle? Because many acids produces inside this. That's why it is known as the organic acid cycle. Okay. Now here, let's study about this Krebs cycle. Actually, it is very easy to remember. Okay, if you studied it properly, so it will be easy for you. Now here, in the starting only I told you that pyruvic acid cannot directly enter inside the Krebs cycle. So these pyruvic acid which convert into the acetyl-CoA, and then this acetyl-CoA enters inside this Krebs cycle. Okay, so this process which is known as the acetylation, and this process which is known as the connecting link between the glycolysis, and this is the Krebs cycle. Now here what happens, whenever this acetyl-CoA enters inside it, it reacts with this oxaloacetic acid. So then there is the addition of this water molecule and it forms the first compound, the first acid, that is known as the citric acid. So this process, this is also known as the condensation. Because here that water molecules get added, and this process, this conversion of oxaloacetic acid into the citric acid, this process which takes place in presence of the enzyme that is known as the synthetase enzyme. And then citric acid is produced here. Then after that, then this citric acid which converted into the isocitric acid. So here 2 steps are involved. The first step, that is known as the dehydration, and the next step is there, that is known as the hydration. During the dehydration, that water molecules get removed out, and during the hydration, this water molecule enters inside it. So whenever from this citric acid, water molecule removed, so that time it gets converted into the cis-aconitic acid. And whenever there is the addition of water molecule into the cis-aconitic acid, then there is a formation of isocitric acid. Then this isocitric acid converted into the oxalosuccinic acid. At that time, the process is taking place that is known as the oxidation. So here, so that isocitric acid converted into the oxalosuccinic acid in presence of enzyme and it converts into the NADH2. At the same time, so that isocitric acid it get converted into the oxalosuccinic acid. Then after that, the next step that is known as the decarboxylation, means what removal of what that carbon dioxide. That step is known as the decarboxylation process. So here what happened? This oxalosuccinic acid undergoes decarboxylation and it forms the alpha ketoglutarate acid. Okay, or you can say that it is known as the alpha ketoglutarate. Now here this process takes place in presence of enzyme that is known as the oxalosuccinate decarboxylase enzyme. Okay, as per the reaction, the enzymes names are there. As per the name of substrate, the enzyme name is there. Now here oxalosuccinic acid undergoes decarboxylation. So here the enzyme is what? Oxalosuccinate decarboxylase enzyme. It is easy to remember. And after the decarboxylation, it forms the alpha ketoglutarate acid. Now, then this alpha ketoglutarate acid which get converted into the succinyl-CoA. If you observe this reaction, so this reaction and this link reaction is same. In this what happens? There is a process of oxidation, decarboxylation takes place. Oxidation, that means removal of hydrogen compound, that hydrogen molecules get removed from this alpha ketoglutarate acid. So this is get accepted by this NAD and it get converted into the NADH2. At the same time, there is a decarboxylation, is what? That removal of carbon dioxide. After the oxidation, decarboxylation process, it get converted into the succinyl-CoA. And here the enzyme which is required, that is known as the pyruvate dehydrogenase enzyme. Here also we have seen the same enzyme, dehydrogenase. And here also this alpha ketoglutarate convert into the succinyl-CoA in presence of that dehydrogenase enzyme. Then here this succinyl-CoA which get converted into the succinic acid. So this state which is known as the ATP formation state. You can see here ATP formation state. Now here there is a formation of ATP. So this succinyl-CoA convert into the succinic acid. So here what happened, that coenzyme get removed and that hydrogen molecule which enters inside it, and then there is a formation of succinic acid. So here the process is of ATP generation. This succinyl-CoA convert into the succinic acid in presence of enzyme. And that enzyme is known as the succinyl thiokinase enzyme. Here the name of enzyme is a little bit different. Okay, that enzyme is what? That is known as the succinyl thiokinase enzyme. So in presence of that enzyme, that succinyl-CoA convert into the succinic acid. Then see that. Then the next process is what? That is known as the oxidation. Means what? From this succinic acid, that hydrogen get removed, that hydrogen get accepted by this FAD and it get converted into the FADH2. See that I have already explained you what is NAD, what is FAD, what is ATP. In the last lecture I have already explained you. So this FAD which accepted this hydrogen molecule and then it convert into the FADH2. Okay. Now here the enzyme which is required, that is that succinate dehydrogenase. Succinate dehydrogenase enzyme is required here. Then this fumaric acid. So here there is the addition of water molecule, means this reaction that is known as the hydration. Because water molecules get added, so due to that this fumaric acid convert into the malic acid, and this process takes place in presence of enzyme that is known as the fumarase enzyme. Okay, then this malic acid get converted into the oxaloacetic acid. Now here you can observe the reaction which takes place, that is known as the oxidation reaction. And here the enzyme which is required for the oxidation reaction, that is known as the malate dehydrogenase enzyme. Dehydrogenase, yani kya? That hydrogen molecule get removed out. So this hydrogen get accepted by this NAD and it get converted into the NADH2. And then there is a formation of oxaloacetic acid. Like this, in this reaction, if you observe here, 3 NADH2 produces here. 1, 2, 3. 3 NADH2 produces. If you observe here, only 1 FADH2 produces. 3 NADH2, 1 FADH2 and 1 ATP, which is produced during this Krebs cycle. Okay, means in this Krebs cycle, you can see, once again I will explain you that reaction. Here condensation, dehydration, then there is the hydration, here oxidation, decarboxylation, oxidation, decarboxylation. Here then there is the process ATP formation, then there is oxidation, here hydration, oxidation. Okay, like this is the reaction which is completed into the cycle. Like this is known as the Krebs cycle. Okay.
Thanks for watching this video. Please like, comment and share my video, and please subscribe my channel. My channel name is Prapti Vanzare. Please press that bell icon.